I apologize in advance for the lack of chairs. I'm told there are more coming. Obviously, I'm very happy to see more people coming to this talk than we anticipated, and I hope your discomfort will be alleviated soon when the chairs arrive, and we'll try and bring them as quietly into the back as possible. Okay, let me welcome you all to the first library lecture for this semester. It's a first, not just in that respect, but also for two other reasons. Firstly, it is the first time that we have had two speakers giving a joint presentation in this series, and I'm very excited about that prospect. And also it's the first time that we have a previous speaker coming back. Professor Tayfun Özçelik was one of our very early speakers a number of years ago, so we welcome him back for that. Today we do in fact have a fairly short schedule. Professor Özçelik has a class to go to after this, plus we have a student workshop in the art gallery at 1:40, so we're going to try and finish more or less on time or a little bit early and then vacate the room. So I shall keep my comments as short as possible. Our speakers, Dr. Katja Dörschner from the psychology department, and as I've said, Professor Tayfun Özçelik from molecular biology and genetics. Their title, Karenina and DNA, Collaborative Aspects of Cognitive Neurogenomics at Bilkent. And they will tell us about a project which has been going on for quite a while now, involving their departments and also people from the engineering faculty, which will be combining work on genetics with brain development and other areas of population development in this region. There's more people coming in as I speak. This is very good and bad depending on how we see. So please, I will stop there. Can you please switch off your cell phones, if possible, to avoid any disruption? And if we have time at the end, we'll have a few questions depending on how we get on. So let me finish there very quickly and then invite whichever of our speakers is starting, Tayfun Bey, to take over. Thank you very much.
Thank you very much for the introduction, David. This is probably the best environment to speak, surrounded by art, and I know that everybody is wondering what's Anna Karenina got to do with DNA in this talk. I'll explain that, but as a starting point, maybe if I tell you a small story, this is the essence of the matter. So we are saying the cognitive neurogenomics group. Actually, the World Congress for Cognitive Neurogenomics convenes, and the scientists come together. And in between the sessions, a group goes out for an excursion, and that place is famous for its hot air balloon excursions. So they take the balloon and they start going over the landscape. First, very nice, very enjoyable. But a little bit later, they discovered that they got lost. And how to turn back, they don't know. So they are looking around, mapping the landscape, charting through, but it's not working. Well, at a point when their hope was diminishing, they just see a group of people walking over there, and they lower the balloon and they shout, "Where are we?" And they shout back, "You are in a balloon." And they said, "Oh, these must be the other guys from the Congress." So we are trying to overcome this. This is a general problem, I'm afraid, in understanding how the human brain works. And the connection with Anna Karenina. Well, actually, the beginning sentence of Anna Karenina is, "Happy families are all alike. Every unhappy family is unhappy in its own way." One of the most quoted opening sentences of novels. Well, actually, this is true for genetics as well. And let me tell you from the beginning, I don't want to go into the technical details. The Genome Project, years and years of research and effort and billions of dollars. Well, we are coming now in 2011 to the conclusion that indeed the genetic disorder in each family may be in a way unique to that ancestry, to that line. So, with these introductory remarks, actually let me outline, because we are now doing a collaboration, I'll give you a brief introduction to genetics, genomics, and genetic diseases, and then Katja Dörschner will talk about cognitive sciences and cognitive neurogenomics and how it relates to our ongoing research projects. I think one of the best starting points is looking at science from a global point of view, and here hypothesis-driven science and on the other hand, big scientific projects that are undertaken by large groups is another approach. In science, I like this quote very much. You can ask Columbus to sail west, but you cannot ask him to discover America. So, in a way, these big projects are like sail to a direction, and then discoveries come along on the way, the unpredictable discoveries. Well, actually, Manhattan Project and making of the atom bomb, the NASA and beginning of the space age, and today we know the particle physics and CERN are prime examples of these approaches. And about, well, now more than 2 decades ago, biology and biomedical sciences also had its share in this approach, and Human Genome Project was initiated, which was completed 10 years ago. Well, many achievements have been made. This is a busy slide, I know, but let me just highlight the main points to you. It was the 1800s that Mendel first became aware of heredity and laid down the foundations of the laws governing inheritance. And then it was Watson and Crick who found the structure of the DNA, which opened the recombinant DNA and molecular biology age. And then in the 60s, how to crack the genetic code, like solving the Rosetta Stone, those were achieved. And then a whole scientific field was unleashed. And this resulted in the birth of the biotechnology industry, as well as understanding the molecular basis of diseases, looking at them at the gene level, plus targeted treatment in the pharmaceutical industry. And thus, in 1990, the Human Genome Project was initiated, which was completed in 2001, the draft sequence and the complete sequence of the human genome in 2003. Ever since, genome projects have gained momentum and have become a central focal point in biomedical research. And we have seen the birth of fields like bioinformatics, and we have witnessed the start and completion of projects like the HAP-MAP, which try to understand genetic variability in our species, and so on. Now, following the completion of the Human Genome Project, the main goal of the scientific community has shifted from doing particular genome projects to sequencing the genome at a considerable less amount, so that all these different types of genome projects can be conducted. And let me give you the figures now. In Moore's law, the quantity of the pace at which a scientific discovery can take place is depicted, and here it is formulated. Well, right now, genome sequencing technology and how it becomes inexpensive far outpaces it. For example, it was $3 billion to start the Human Genome Project. About a decade ago, it was $500 million to sequence a genome. In 2007, Watson's genome was sequenced as the first human being from the beginning to the end. It was about $1 million. And last year, when we started our whole genome sequencing in the context of the project that I'm going to tell you today, it was about $50,000. Right now, it is $10,000. And the goal is to bring it down to $1,000 by the year 2014. If you imagine that only 1.5% of the human genome is encoding genes, then gene-related parts would be sequenced less than $10. So this is actually a far-reaching development. And it's not like a tsunami that comes on us, but it's going to be a gradual tide rising and rising and rising and dominating the whole field of evolutionary studies, understanding the function of the genome, disease-based studies, and of course cancer. So in fact, these are the examples of projects that are being conducted at present in different laboratories around the world. It is predicted that, well, if you divide the decades down here, like 1990s to 2003, 2004 to 2010, from 11 to 20 and beyond 2020, though it's difficult to make predictions for 2020, I understand that, the focus begins from understanding the structure of the genome, this is sequencing the genome, to understanding the biology of genomes, model organisms, to understanding the biology of diseases, which we are now living this era, to advancing the science of medicine, and we hope that this will result in treatment modalities for patients within the next decade. So within this complexity, what we have learned, as I said in the beginning of my talk, is that genetic diseases display extreme heterogeneity. By heterogeneity, what I mean is locus heterogeneity, different genes are involved. An example, deafness. Deafness is associated with at least 150 different genes in the human genome. Today we know this. Or mutation heterogeneity, meaning along a gene there are many different mutations that may give rise to different phenotypes. This may sometimes complex to the level that a mutation in one portion of the gene gives rise to a particular disease, and in the other part of the gene it may give rise to another disease. Example, testicular feminization. This is a famous disease. They look like females, actually also beautiful, but secondary sexual characteristics do not develop. Why? Because the androgen receptor gene is mutated. So since there is no target for testosterone in the body, during development the secondary sexual characteristics don't develop. The same gene, this is the portion for hormone binding domain of the gene. When the hormone binds to the gene, it goes into the nucleus and activates a set of genes with a domain that is capable of binding to the DNA. Now, if the mutations are in the DNA binding domain, the result is what's called as Kennedy disease, or spinal bulbar muscular atrophy, in which the patients exhibit a specific spinal and bulbar atrophy of the muscles, so they have walking problems and so on. So how are we going to solve these challenges at the genomic level? Well, of course, this is achieved by conducting genomic studies in families that are afflicted with these disorders. And we analyze their genome and we try to find out where the faulty gene is. And in this type of research, actually, if you look at the world, and when you see this map here, you'll first now remember, I think, Libya, and then its commodity, oil. Well, actually, from a scientific point of view, in this region, extending from the southern shores of the Mediterranean Sea, the northern Africa, going through the Middle East, of course, Anatolia, all the way to India, is consanguinity. This is, I may dare to say, a commodity that is most valuable to understand the human genome. So we want to take advantage of this human tragedy, but a scientific opportunity, and start dissecting the molecular basis of inherited disorders, because if you find the genes that are associated with these disorders, then this gives you the chance to identify the normal proteins, normal pathways that are important for the development of those structures in the human body. So, in fact, one disease that we studied is the quadrupedal locomotion in man. Unertan, a physiologist at Çukurova University, discovered this phenotype in 2005 and caused a lot of actually controversy and discussions in the world in all scientific grounds and popular press. But this was indeed a very interesting phenotype for us, and this actually existed on this land for many years. This is expected, but we were lucky enough, thanks to Cemal Yalabık, I am indebted to him. He saw this book, Across Asia Minor on Foot. It's a book by W.J. Childs. He was at the California University at the time, and I think a missionary in Anatolia in the early part of the 1900s. And in 1915, he was at this part of our country, and one day when he was going to Baghdad, he said he saw this man and took a picture of it. He says, I was walking on the road, and then a most interesting human started walking towards me and then asked for money, and then he describes the beggar of Baghdad. So because it's on the way to Baghdad. So this is the earliest case that we can find with quadrupedal locomotion in humans. Why quadrupedal locomotion attracts so much attention from the scientific circles in the world? Because understanding human evolution, the genes that have been associated with the development of our brain, enlargement of our brain, our ability to talk, our ability to use tools, and so on, there are specific diseases that are impaired. So if we can find a mutation that hits a particular gene, then we have the chance to understand the pathway. After Darwin, the missing link studies was conducted throughout the world. It still goes on. And what are the critical steps in human evolution has been also one of the most disputed and heavily studied areas. Now, the scientists in the 1800s up until 1950s thought that it is the enlargement of the brain that triggered our species to diverge from the lower primates. Here is the picture of Lucy, the first one of the earliest primates that's diverged into the Homo sapiens group. But then it became clear that brain enlargement is a later event and that there must be an earlier one. Tool use was thought to be the critical event, but since 1970s, almost everybody working in the field of evolution agrees that bipedal locomotion, our ability to stand up and free our hands from the ground, was the earliest event. That way, the hands are free, they are no longer ruined, they can do fine tasks, which triggers, of course, the enlargement of the brain, and so on. Of course, it's not a singular thing. It requires also the environment and what they eat, what they drink, and so on. But that's why bipedal locomotion attracts attention. And what we have seen analyzing this genetic disorder is that here in this picture you have the families that are afflicted with this disease that come from regions like Istanbul and Çanakkale and Afyon and Adana, Hatay and Gaziantep and Kars and Baghdad, on the way to Baghdad somewhere. The diseases, this is such a rare phenotype, though there is extreme genetic heterogeneity. Almost in every family there is a different chromosome that is involved. And here 4 of them are shown: chromosome 9, chromosome 17, chromosome 13, and chromosome 8. When we analyzed the chromosome 9 gene, we found that mutations in the very low density lipoprotein receptor cause the phenotype in these humans. Well, actually this was a most unexpected finding, because very low density lipoprotein is the closest relative of high density, the good cholesterol, and low density lipoprotein, the bad cholesterol. And there is the third sibling, very low density. These genes were discovered by Brown and Goldstein in the late 70s and 80s, and they were awarded the Nobel Prize in 1985. And subsequently, they have built up a large group in Texas, and ever since have been studying the biological functions of these genes. And along the way, very low density lipoprotein receptor was a real mystery, because when knockout mouse were produced, for example, no phenotype was observed. In fact, very low density lipoprotein receptor, now today we know, is a receptor for reelin. This is a protein that is important for the development of our cerebellum. It serves as a receptor for this protein, and that indeed human phenotype is cerebellar atrophy. This was an editorial, part of an editorial in Nature Genetics at the time the discovery was made, and at the same time this was the first example of an identification of a disease and finding its gene in a country from the Mediterranean basin. In fact, only Turkey, Israel, and Italy are regarded as those that are capable of doing such studies, and became again another editorial in Nature Genetics, basin of attraction, and a paper. Well, then we turned our attention to the second gene, and when we sequenced the family, actually this is whole genome sequencing, we found a mutation in a gene that has never been characterized before. And this resulted in my student, I think here, Süleyman is here, he received the Isabel Oberle Young Investigator Award of the European Union. I think the one of the most prestigious awards that are given to young scientists. Well, this is not enough. Actually, all your competitors are also very clever, and they are finding ingenious ways to beat you. And nowadays, genetics alone is not enough. It is necessary to study human, study model organisms like mouse or zebrafish, study the cognitive function, look at the brain, do imaging studies, and paint the complete picture. So, and we have been very lucky here at Bilkent because our interests, Hüseyin Boyacı, Katja Dörschner, Ergin Atalar, Michel Adams, and Ayşe Tekinay, and myself merged, and we call ourselves the Imaging, Genetics, and Neurosciences group. And we are starting to understand the brain beyond its gene. The very first example, I think this is one of the last slides that I'm going to show you, is the mouse studies that we have done. The new gene that we have identified, we've seen that it is highly expressed in the brain, shown up there, and in the mouse, the localization of the brain is to the Purkinje cell layer of the cerebellum, which is actually a very supportive finding that the gene is indeed involved in the development of the phenotype. I'll skip this. So I think what Arthur Kornberg predicted in the early 70s is coming true. He said this 20th century biomedical sciences can be divided into phases, and the beginning part is the microbe hunters. People were discovering new microbes, and these were the most significant scientific discoveries. Then came the age of vitamin hunters, vitamins C, K, B. These were different types of Nobel Prizes at that time. In the 40s to 60s, gene hunters were on the stage, which eventually led to the genome projects, and in the 21st century, understanding how the brain works is going to be a major challenge, and he calls it the headhunters. And this is further reiterated by significant people in the field, that marriage of genomes and neural circuits, it is time to do that. So with these remarks, now I would like to leave it, and now we are, and how we open Pandora's box to understand the brain, to Katja. Thank you very much.
Okay, I'll deal with the subtitle of the talk, Cognitive Neurogenomics, the marriage of two fields of research. So, how does the mind work? That's one of the most interesting questions to ask in science. And cognitive neuroscience, cognitive neurogenomics, is a new emerging field which studies or tries to establish the link from gene to brain to complex behaviors. As you see in the pedigree here, cognitive neurogenomics has its roots in two disciplines called cognitive neuroscience and genomics. Cognitive neuroscience studies neural substrates that subserve cognition, and it has its roots in neuropsychology, cognitive psychology, and computational modeling, whereas genomics is rooted in genetics and molecular biology. Now, historically, the question of trying to understand the mind has been to try to localize the faculties of the mind to certain structures of the brain. So let's look a little bit into the ancestry of this field, and instead of starting with Aristotle, who apparently did everything first, I'm going straight into the 18th century and the great-grand uncle of cognitive neurogenomics, which is phrenology. Now, phrenology, as some of you may know, tries to study the mind by feeling out the morphological differences on the skull. The idea being somehow the shape of the skull reflects the underlying shape of the brain, and if you're a hard thinker or a very emotional person, that part of your brain has been put to work a lot, and hence that will reflect in the shape of the skull. Now, of course, we know that that idea is pseudoscience. However, the important main idea to take away from this is this idea of functional localization. So a certain faculty of your mind is localized to some part of your brain. Now, if we move down the history line and encounter the grandfather of cognitive neurogenomics, we have neuropsychology. Neuropsychology tries to understand the cognitive functioning of the healthy brain by studying lesions, so lesions in patients, people with brain damage. Two of the forefathers or pioneers in this field are Paul Broca, French physician and anatomist, and Carl Wernicke. Broca studied patients who suffered from aphasia, so an inability to utter language. And by looking at their brains, he found that in fact they had lesions here in the premotor areas, inferior over here, on the left side. And he concluded, all right, I found the center of language processing. So, and it's interestingly now it's called Broca's area. He said, interestingly, by making this claim, he proved phrenology wrong and right at the same time. He proved it wrong because Franz Gall thought language was, I don't know, in some other part, like frontal part of your brain. But Broca showed that that is not the case. On the other hand, this is the ultimate scientific evidence that there really is functional localization in the brain. Now, 20 years his junior, Carl Wernicke, thought that that claim that Broca made was a little bit bold to have found the language center, because he noticed that there are patients which are fluent, but have a different kind of language deficit. So they don't have this agrammatic speech. Patients with Broca's aphasia, they could only utter words like yes, me, go, no, and had really problems expressing themselves, but could comprehend speech, whereas patients who had lesions in this area, highlighted it here in blue, were very fluently, grammatically speaking, but it didn't make any sense whatsoever. So he thought language is a more complex phenomenon than it can be just going on in one area. And he proposed in his book, which he wrote like in his mid-20s, Der aphasische Systemkomplex, he hypothesized that language is a very complex system and many cortical areas involved. In fact, by now we know it's not just Wernicke's area and Broca's area, but there are people who have lesions in within these regions, and they have very interesting deficits, like they can utter nouns, but they cannot utter verbs. So very specific, interesting deficits, which tells you that language is more complex than just two areas. In any case, this quest for functional localization has been carried on into the present days, and cognitive neuroscience tries to, or has now the technological means, for example, by using magnetic resonance imaging, to look at the functioning of the brain in healthy, so that is non-brain damaged people, and to localize function in the healthy brain. So here I show you one example there. So here you can see that is the cortical activity when a person is engaged in a language production task, and what you would expect that the region identified by Broca should be at work, and this is exactly what we see. So we have the means now to study functional localization in healthy and also in lesion patients. Now, why did the marriage happen between cognitive neuroscience and genomics? Well, to make it short, the time was right. In a 2007 Science article, a group of very prominent researchers proposed the time is almost there. Like, we have the technological means to understand how the brain perceives things and acts, and we have to put a major effort in the next decade to try to figure it out. Ironically, what this means is that we haven't figured it out yet. And why have we not figured it out yet? Well, it turns out the brain is an incredibly complex structure. You have hundreds of billions of neurons, and these are all interconnected with each other in one way or another. Moreover, there are more than 2,000 genes, probably far more. My source was from 2004. Expressed in the human brain, which tells you that there's also developmental. It's a very complex structure that we're dealing with. The second reason, as you already heard today, is the completion of the genome project. And this rapid and high throughput sequencing and interpretation of entire genomes makes gene discovery and gene mapping feasible today. Lastly, the advantages in neuroimaging, like functional MRI, diffusion tensor imaging, which lets you get very detailed, accurate pictures of the brain at rest or at work without having to open any skulls, is a third reason why this is a good time to merge these two fields. Now, we propose that to truly understand the mind, that must involve the identification of the genetic mechanisms that contribute to complex behaviors. Why is that? You saw this article already before, but a group of Nobel laureates and very esteemed researchers proposed that it is really high time now to merge the efforts in terms of the methodologies of the fields of genomics and of cognitive neuroscience in order to understand, treat and prevent, for example, psychiatric illnesses. So this is not a contrived synergy, it's a really needed thing right now. The cognitive neurogenomics addresses this call that's forwarded by this article, and that is this kind of integrative neural science. One way that cognitive neurogenomics accomplishes this is by relating genomic and neuroimaging data. Now, let me tell you about the three major routes that this can happen. One way to relate these two kinds of data is by doing twin or pedigree studies. So what you do here is you're trying to establish the heritability of human neuroanatomy, like gray matter volume, for example. So, and the cortical function of the variability that we see between cortical structure of people in the brain is attributable to genes. And here's an example from Thompson and colleagues. In this column you see identical twins, data from identical twins, and here are fraternal twins. So these share the same set of genes, hence identical, and these are like any other siblings, share some but not all. And what is shown here is a correlation, how much the gray matter volume in this case correlates between identical versus fraternal twins. And you can see there is really high correlation, especially in the frontal areas of the brain in terms of gray matter volume. This is very interesting and makes sense because we know from the studies of extended families which are have relatives with schizophrenia, that they share similar, the unaffected individual in those families share similar cortical structures in the frontal parts of the brain. A second way of associating genome and imaging data is to do genome-wide associations. That's a massive computational effort because you're taking several candidate genes or single nucleotide polymorphisms, so functional variations of the genes, and try to link them to all possible parts of the brain. Like it's actually, we're measuring this in terms of voxel. I don't want to further go into this, but this is the unit that we're dealing with neuroimaging data. So they're correlating voxels with lots of lost and single nucleotide polymorphisms. And lastly, the third approach is that if you use illnesses where you have already the genetic basis identified, and you have some candidate genes that you're postulating that may be playing a role. So this is the most direct method to study the effect of single genes on the cortical structure and function. And in fact, the collaborative project that we've been doing together here at Bilkent falls into this third category. So let me briefly tell you about two projects. I'll start with quadrupedal locomotion. You've already heard about the phenotype, what these people are like. Other than their quadrupedal walk, they also have real problem with language, like uttering language, expressing themselves, and of course mental retardation. We invited the family from Hatay to come here for 2 days and then to do extensive measurements on them. So what we usually do for such a measurement, so here you can see our 3 Tesla magneton trio scanner at Umram, and there's our postdoc putting, this is not a patient, but another student from the center, into the scanner. So the person goes into the center of the board, and then we're running scans to obtain structural and functional data from these patients. So for this particular group, we measured the activity of the brain during rest, because we had to sedate them mildly, because as they are mentally retarded, it would be too stressful to go into this confined space. But you can still measure cortical activity at rest, but I won't talk about those data today. We also did diffusion tensor imaging, which allows you to visualize the white matter trajectories in your brain. I'll talk about that in a moment. So here we have the family, and you see here 4 affected individuals. So these carry the mutation, and here you have 1 unaffected individual, a sibling, and a carrier. Now you can try to be a cognitive neurogenomicist and maybe just by eye try to understand what's the structural differences between these 2 and these 4. You probably remember from what Tayfun told you already, their cerebellum is highly atrophic. So one thing, you can see their cerebellum, the structure over here, is very small compared with the healthy individual. Another thing that stands out is that the corpus callosum, so the white matter structure which connects your left side of the brain to your right side of the brain, is very thin in these individuals. But that's not science, right? Looking at pictures, telling this is big, this is small. So we want to do this quantitatively. And we basically, this is an analysis of morphometric features. This work was painstaking effort done by our postdoc, Aslihan Ors. She used the software FreeSurfer, which can compute several indices of cortical structure. So one is, for example, cortical thickness, how much your gray matter varies. Gray matter and white matter volume, as like depending on the area where you are, or mean curvature, so a measure of gyration. And if you have, let me just tell you, if you have more cortical thickness does not necessarily mean that's a good thing. Any deviation from normal in these measures is usually something bad. There is an optimal level of cortical thickness, volume, and mean curvature, and so on. Now, we compute these measures on our patients and on a group of age-matched healthy controls, and see where there are significant differences between the patients and the controls. Let me show you some results. What you see here is a sagittal slice, so these are structural MR images of a control, so an affected healthy participant and a patient. And you see here, highlighted are the regions which show volumetric differences. And as we already saw by eye, so good thing, it's not so bad, there are actually significant differences in the volume of the corpus callosum. So it's much smaller for a patient than for the control. Also, as we already noted, there is a difference in the volume of the cerebellum, and also differences in the ventricles, so fluid-filled spaces in your brain, like third and fourth ventricles. What you see in panel B here is, it's a little hard to see, but it's an inflated cortex. So it's a 3D graphical rendering of what the brain should look like when you would take it out, but we can do this without having to open the skull. And here you see cortical area highlighted, which show difference in these other measures that I mentioned, like curvature, mean curvature, volume, and so on. And I just want to draw your attention that the areas which show significant differences, so this is a lateral view, you're looking basically on this side, and this is the medial view, we're cutting the brain in half and you look that direction. These are motor areas, areas that play a role in your ability to send signals to your muscles that they should move. And also here, you may recognize by now that this is roughly where Broca's area is located, which may explain their ability with their problem with the ability of speaking, and also may explain the problem with their gait. So these differences in motor cortex and also cerebellum. So cerebellum has been known to play a very important role in your ability to maintain balance and walking and so on. Now let's look at the diffusion tensor imaging results. So the diffusion tensor imaging measures water diffusion in your brain, and what happens is that in white matter, so where the axons of your neurons, that diffusion is highly directional, and what that allows you is to reconstruct the 3D trajectory of white matter pathways. So here we see again a sagittal slice through the brain of patients and two control subjects, and we here try to show the trajectory of cerebellar peduncle, so the white matter pathways which go from cerebellum to cortex. There are 3 of them, and you can see, maybe it's best illustrated here, that these are very also atrophic in the patient compared to the control subject, so it seems much less complex. So another candidate that may explain the phenotype that we are observing. Let me briefly talk about a second ongoing project, a patient with occipital pachygyria. We were in a very fortunate position to be collaborating with Murat Günel at Yale University, who had been studying people with Lissencephaly syndrome, trying to identify the gene that is responsible for that. And he wanted us to do some structural and functional investigation of this patient. So the patient was a 34-year-old female and high school graduate, and when you met her, she seemed perfectly normal. Are we running out of time, by the way?
2 minutes.
2 minutes. All right. I will skip this one and go straight to the conclusion. So we'll have to do another talk if you want to learn more about this. So what have we learned? By combining the efforts from genomics and cognitive neuroscience, we're beginning to understand what genes might be involved in cortical structure and or function. So these are the very first steps. This, of course, works better if we pair it with comparative genomics and try to study the expression of genes throughout development, which Tayfun mentioned is already also ongoing in our group. And I would also like to highlight the value of discovery-driven science. So this is very different than the way we normally conduct science, which is in cognitive neuroscience, which is mostly hypothesis-driven. But clearly, this approach proves the value of discovery-driven science, very much to the analogy that Tayfun was given earlier with Columbus sailing to West versus Columbus sailing to discover America. So, in conclusion, Aiming to uncover the link between gene, brain and cognition, cognitive neurogenomics not only carries on the quest for functional localization that had begun with the lesion studies of Broca and Wernicke, but also has the potential to shed light on the factors leading to the evolution of the capacities and faculties of the human mind. I apologize for this very German sentence, and I would like to highlight just the contributions of Burak, who is doing the resting state analysis, Aslı, who is doing the morphometry analysis. She's a postdoc in our lab. He's a master student. Hüseyin Boyacı, who's involved in every aspect of the project, and Ergin Atala, who has been supportive of this project from the beginning in his capacity as a director of UMRA. So thank you very much.